Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar titled Transforming Viral Vector Manufacturing with the Next Generation CGMP Plasmid Platform. I'm Abby Pinchbeck, an editor at BioInsights, and joining me today are John Bowen and Brian Tomkowicz, who will explore innovations in CGMP plasmid production that enhance viral vector manufacturing by improving efficiency, scalability, and cost effectiveness, while ensuring consistency and reliability in cell and gene therapy development. After the presentation, if we have time, we'll have a live Q&A session. So we invite our audience to pose their questions to our speakers using the Ask a Question box at the bottom of your screen, and we'll try to get to these during the session today. I'd also like to draw your attention to the resources on the right-hand side of the screen, where you can find more information on the topics covered today. Now I'd like to introduce our presenters. John Bowen, Senior Director of Nucleic Acids and Plasmid Operations at SK Pharmtechco, has over 10 years of biopharmaceutical experience, including roles at Novartis and Merck, with expertise in viral vector development and plasmid manufacturing. Brian Tomkowicz, Senior Director and Head of R&D at SK Pharmtechco, is a cell and molecular biologist with over 15 years in biotech, specializing in viral vector engineering and gene therapy development, with prior roles at Janssen and UPenn. So without any further ado, I'll hand over to Brian to kick us off with the first presentation.
Thank you, Lauren, for the kind introduction, and thank you, BioInsights, for hosting this webinar today. I'm Brian Tomkowicz, Head of Research and Development at SK Pharmtechco US, located in King of Prussia, Pennsylvania. Today, we'll be discussing the importance of plasmids in the viral vector manufacturing process. SK Pharmtechco's mission is to support its partners in delivering life-saving therapies by leveraging cutting-edge technologies while maintaining a strong focus on customer needs. Our vision is to be the most trusted global partner in delivering innovative medicines to patients and stakeholders worldwide. We are proud of our commitment to making a meaningful impact on patients' lives globally. SK Inc. is a South Korean conglomerate and the parent company of SK Pharmtechco. SK Inc. has built a strong foundation of operational excellence across a wide range of industries, including energy and chemicals, semiconductors, telecommunications and IT, biopharmaceuticals, and life sciences. It is committed to being an industry leader with an emphasis on environmental, social, and governance through continuous investment in eco-friendly projects and technologies. SK Inc. has remained a stable business by emphasizing a strong vision for success through global leadership, innovation, sustainability, and customer-centric growth. It continues to expand its global presence through investments in new technologies to ensure long-term responsible growth. SK Pharmtechco is a contract development manufacturing organization with a global presence across North America, Europe, and Asia. As part of the SK Pharmtechco organization, we offer multimodal capabilities for small molecules, cell and gene, viral, as well as plasmid manufacturing with compendial analytical testing to support the pharmaceutical and biotechnology sectors. SK Pharmtechco operates facilities in key locations such as the USA, Ireland, France, and South Korea, focusing on small molecule manufacturing, process R&D, and analytical services. Our cell and gene therapy manufacturing centers are located in Silicon Valley, located in the suburbs of Philadelphia, and facilities located outside of Paris, France. Each facility is tailored to support specific aspects of pharmaceutical manufacturing and research, further reinforcing our global reach and expertise. Plasmids play a crucial role in the manufacturing of advanced therapies. They have wide-ranging applications from starting material to drug substances. Plasmids are defined as DNA vehicles used to clone, amplify, and express genes in target cells or tissues. They are essential in therapies such as DNA and RNA and protein therapeutics, as well as critical drug substances for use in the manufacturing of viral vectors and gene-modified cell therapies. I briefly summarized the applications and stages of maturity in the field, emphasizing the evolving and established technologies in clinical and therapeutic settings. At the onset, plasmids play a foundational role in viral vector production, as plasmids serve as templates for creating essential viral components during manufacturing, whether they encode the actual therapy itself or the structural genes required to make virus. The quality, structural fidelity, and functionality of these plasmids are critical factors that influence vector yield, stability, and safety, all of which are paramount for clinical applications. Our unique service offering in early-stage plasmid development focuses on the pre-production or design phase. We place a strong emphasis on several critical components prior to manufacturing plasmids for your program. This includes a comprehensive annotation and plasmid design strategy to ensure the expected function aligns with the actual design of the plasmid. Often overlooked and otherwise assumed to be correct, we perform this crucial early check to be sure that all intended elements of the plasmid are expected based off the design strategy. For cell and gene therapies, the gene of interest is flanked by complex DNA structures required for viral packaging and sustained expression of the therapy upon infection. The inverted terminal repeats, or ITRs for AAV, and long terminal repeats, or LTRs for LVV, are complex sequences that can often be subject to mutation during cloning and plasmid scale-up. We can assist with designing your plasmids to maximize performance by offering a comprehensive suite of regulatory elements such as promoters, poly-A tails, insulators, and other regulatory elements, as well as species-specific codon optimization to ensure high expression of your cell or gene therapy. Through the Compendium of Plasmid Science, early to late-stage PD process establishment, as well as through GMP manufacturing, we carefully monitor sequence integrity by using high-fidelity next-gen sequencing approaches. This ensures the highest quality raw materials to support robust manufacturing. We'll spend the next few slides discussing plasmid sciences in gene therapy. Illustrated in the diagram to the left is an example of a packaged genome of a recombinant AAV gene therapy. Contained within the virus is the DNA encoding the promoter, gene of interest, and regulatory elements flanked by the viral ITRs. The closer inspection of a typical sequence that harbors the AAV cis plasmid or gene of interest shows the components that make up the plasmid. Through our ongoing research efforts, we have discovered that the orientation of the components that make up a plasmid, such as the ORI, antibiotic resistance gene, and viral elements, can influence the frequency of mutations in the ITR regions. These mutations can be as simple as a point mutation or a large deletion, and based upon the design, we can predict where these mutations will occur in the plasmid. By understanding how these design features increase risk through plasmid scale-up, we have developed strategies to completely eliminate the mutations in order to maintain plasmid stability through all stages of manufacturing. Complex viral promoters are also hotspots for mutations, given that these sequences often harbor large GC stretches of DNA. They can frequently be spliced out during packaging of DNA into viral capsids. Using next-gen sequencing of purified viruses, we can monitor the location and frequency of these unintended events to enable continuous improvement of your gene therapy. As shown in the bottom panel, we offer validated and fully sequenced plasmids as off-the-shelf product offerings for use in your gene therapy project. On the bottom left panel, we design plasmids that not only compete with competitor plasmids, but have also been modified to eliminate the formation of replication-competent AAV in your drug product. By maximizing performance and rearranging the P5 promoter, we can not only maintain peak performance, but also simultaneously ensure safety. Lastly, through a production process of an AAV9 purified virus comparing our helper plasmid to a competitor, we show excellent performance of our P-helper design. This figure shows the mass balance recovery at each process step as measured by digital PCR at harvest, affinity capture, and anion exchange polish. This slide explores various AAV cis plasmid designs that vary in length and promoter complexity. It includes not only complex viral promoters such as CMV, but also tissue-specific promoters for liver, muscle, and neurons. The left graph provides a schematic representation of the various cis plasmids used in the study. All sequences were confirmed using next-gen sequencing. Next to the schematic diagrams are the indicated lengths of the expected packaged genomes. The first plasmid, designated with the PGK promoter, is a self-complementary AAV intended to be less than 2.3 kb, while all the other plasmids are for normal packaging sizes up to 4.7 kb. In the middle, we show the production titers for both AAV2 and AAV9 plotted as a function of the genome size. For this experiment, we utilized our proprietary HEK293 clonal serum-free suspension cell line, 4G9, which is currently in use for all viral vector manufacturing at the facility. As you can see, appreciable titers were noted, irrespective of packaging size or complexity. The graph to the right is the same plot as the center. However, we labeled the x-axis with the actual cisplasmid ID. For both AAV2 and AAV9, excellent titers were noted, with AAV9 titers consistently higher, as expected. I'd like to spend a moment to discuss the importance of early-stage plasmid design and annotation. The first case study was a request to convert an ampicillin to kanamycin-resistant plasmid for use in manufacturing. Sequence analysis of the plasmid, coupled with detailed annotation, revealed an unexpected mutation in the promoter region. This mutation altered the tissue specificity of the gene expression upon infection. Unbeknownst to the client, this was not intended. We were able to carefully map the location of the alteration to a non-canonical restriction enzyme not typically found in promoters, which was introduced somewhere during the development of their program. The DNA that followed this restriction enzyme site altered the tissue specificity of the promoter. Through these critical first step efforts, we were able to provide valuable information to the customer to enable them to make an informed decision on how best to proceed prior to investing in costly plasmid manufacturing. The second case study illustrates the importance of understanding how plasmid design impacts process parameters during scale-up. As with any process, maximizing speed, yield, efficiency, and accuracy is important. However, in the context of plasmid scale-up, it's critical to understand which of these variables can be modified to couple speed, yield, and efficiency with accuracy. By coupling next-gen sequencing along with restriction enzyme digest analysis, throughout all stages of plasmid manufacturing, we were able to identify process parameters that negatively impacted the production of a very complex GC-rich plasmid. These parameters include bacterial strain selection, incubation time, and temperature. By understanding how all these parameters impact each other, we were able to successfully create a unique production process to mitigate mutational risk while still meeting the plasmid demands for the project. Through our ongoing research with a focus on creating sustainable manufacturing by seeking opportunities for cost of goods reductions, we leverage our available plasmids to streamline the viral manufacturing process. Typical AAV production relies on triple transfection of P helper, the structural rep cap plasmid, which defines tissue tropism, and the cisplasmid, or gene of interest. By combining the rep cap plasmid onto our P helper plasmid, and thereby converting to a double transfection process, we were able to show similar titers for an AAV2 produced virus. The downstream impact of this modification would enable the customer to lower plasmid costs by approximately 1/3, since only 2 plasmids would be required during manufacturing. The graph on the left illustrates similar titers between triple and double transfection. However, the incorporation of a viral enhancer during production can significantly improve titers, as shown in the center graph. The graph on the right highlights the fold change in titers between the two platforms. Taken together, these subtle changes can have a profound impact on cost of goods reductions. If we examine how these viruses perform in vitro cellular transduction assays, focusing specifically on triple versus double transfection produced viruses from the previous slide, we see quite a stark difference in potency. The AAV2 virus used in this example is a dual reporter encoding for both GFP and the firefly luciferase genes. Shown in the left figure is a comparison of GFP expression at decreasing multiplicities of infection from 1,000,000 MOI to 8,000 MOI, while the graph on the right measures luminescence or activity of the luciferase gene. The double transfected produced virus outperformed its triple transfection counterpart. We provide these examples to demonstrate the power of plasmid engineering as potential alternative manufacturing solutions. These are available reagents for use and can be tested to determine if they might enable cost of goods reductions while maximizing productivity for your program. I'd like to turn the presentation over to John Bowen, who will now provide a detailed overview of the plasmid manufacturing process. John?
Thank you, Brian. So in addition to the plasmid science, a great amount of effort was done to establish our manufacturing platform. This platform development encompassed 3 main manufacturing objectives. 1, to maintain a high-yielding, reproducible, and robust process that can accommodate a variety of plasmid types, sizes, and sequence complexities. 2, to maintain plasmid quality. Essentially, this means to enrich the supercoiled plasmid isoform while minimizing the formation of and subsequent clearing during the downstream process of any unwanted DNA isoforms. Typically, this includes isoforms such as linear, open circular, and any residual E. coli DNA inherent to the E. coli process. And finally, to clear any other process or product-related impurities such as host cell RNA, host cell protein, endotoxin, or anything else inherent to your product or process. Also, we demonstrate a consistent panel of compendial assays and other residuals outside of residual host cell protein, RNA, and DNA to maintain consistent quality and performance across our platform. A high-level process flow description is shown on the bottom of the slide. So each unit operation is shown, and it begins with a high-density fermentation to propagate our E. coli cell mass and increase the plasmid copy number per cell as a function of time. Next, the cells are harvested via a high-speed centrifugation to separate the cell pellet and intracellular plasmid from the fermentation supernatant. Upon the plasmid being liberated into solution, a series of flocculation and neutralization steps are occurring. Now, these steps are very critical to ensure that any residuals and debris are flocculated and precipitated out as much as possible before entering the downstream process. This provides a physical barrier and becomes filtered out prior to loading a high endotoxin, HCP, or RNA load into your downstream process. These midstream steps, such as the resuspension, lysis, and neutralization, are very prone to plasmid degradation, and this is primarily due to the harsh acidic and basic conditions that occur during the lyse. So a platform is very critical to ensure batch-to-batch reproducibility and minimize any product loss, as even a few minutes at any one step longer than the critical processing time can lead to dramatic yield impacts at this stage. To remove any of the unlysed cells, cell debris, and flocculated residuals, a clarification step is then performed prior to the downstream unit operations. The downstream purification step is governed by a two-phase chromatography process. The first is an anion exchange capture chromatography step, where pDNA is separated from RNA and other process-related impurities. This is followed by a hydrophobic interaction step, which serves as a polishing operation to further remove any impurities. The Hicks step is the main unit operation to separate the supercoiled DNA from any open circular plasmid as well. The plasmid is then finally concentrated to a desired concentration via tangential flow filtration and undergoes a final sterile filtration step into a container of interest as dedicated by the client. In addition to establishing the plasmid manufacturing platform, particular care was spent on selecting the proper E. coli strain and particular clone for each new plasmid that we receive. So this was done during our initial platform development using 3 different E. coli strains, 4 different representative plasmids of varying size, sequence complexity, and use in a typical CGT process. And what this means is we chose a subset of particular AAV and LV plasmids to use in our initial platform design. So in addition to doing this for our platform stand-up, we also execute this for each new plasmid that a customer brings in our hands. So we demonstrate that it's compatible with our process. We evaluate it in our clone and strain selection, and we offer any technical consulting services as part of our plasmid science and evaluation. So this slide indicates a case study showing how important it is to select the proper E. coli strain and colony and how it impacts fermentation identity and fermentation yield. So multiple strains of E. coli were transformed with a representative plasmid, plated on LV plates, and then allowed to propagate overnight. So specific selection criteria was included, such as colony size and incubation temperature, and these parameters were screened to then select the best clone for each individual strain. We also looked at different colony sizes as well to determine if that had any impact on the sequence integrity. In this instance, we were looking at ITR containing plasmids for a gene of interest in an AAV vector platform. So each strain was placed in our high-density platform fermentation process and harvested. At the lower part of the slide, you can observe both the difference in yield, where we ranked qualitatively as high, medium, and low, and the sequence identity, ranging anywhere from 91% up to 98% matching to the reference sequence. So this indicates how important not only the high density fermentation process is, but how important it is to screen each plasmid strain and clone prior to beginning a GMP large-scale manufacturing run. So without going into specifics on our fermentation platform, we wanted to share some data and how we developed the flexible and scalable plasmid platform. So as part of our upfront development, like I talked about on the last slide, we chose 3 different strains and 4 different plasmids with varying attributes. So we chose common AAV and LV plasmids that varied in size, sequence complexity, GC content, and even looked at different promoter and poly A tail sizes and different elements in the plasmid backbone. So primarily, we looked at this with any ITR or LTR containing plasmid, figuring that was a worst case scenario for any AAV and LV process, respectively. So some of the critical process parameters that we looked at are listed at the table below in the lower left-hand corner. So these include E. coli strain, fermentation temperature, the temperature profile throughout the run. And essentially what I mean by this is, do we maintain a constant temperature? Do we increase or decrease the temperature at any given time to give some sort of temperature shift to increase the cell productivity from more of a plasmid amplification and less of a cell mass biogeneration state? We also looked at other common fermentation parameters such as temperature, pH, and dissolved oxygen. And what this did is this allowed us to determine the impact of each of these critical process parameters on both yield and quality to set strict ranges for each of the plasmid types that we would see going forward. So other than your typical processing parameters, we also spent a fair amount of time on selecting certain media and optimization of this media components. So looking at the proper carbon source, so is essentially looking at glucose, glycerol, or other carbon sources, looking at an animal origin free or a completely defined versus semi-defined media. These were a lot of the things that we looked at in our upfront development as well, which we looked at what's important to our customers and then what gave us the best productivity in our process. So in addition to doing a base media screening, we also looked at a feed media as well. So typically a little lighter and geared toward switching the plasmid production from cell growth into more of a plasmid amplification phase. So in addition to changing the media composition in our feed, we also looked at a different strategy based on the plasmid type. So for example, larger plasmids with more complex sequences may require a different feeding strategy, a different temperature profile, as what we would typically do for a smaller, less complex plasmid. But either way, regardless of the fermentation parameters that we've developed for both a small, less complex, and a larger, more complex plasmid, we've observed very consistent cell growth and plasmid productivity across a number of batches, as we can see in the OD600 chart shown in the lower right-hand column of this slide. As mentioned previously, anion exchange chromatography and hydrophobic interaction chromatography are really the workhorse of pDNA purification. So due to the negative charge of both plasmid DNA and RNA, proper development of the wash and elution conditions on the anion exchange column needed to be developed up front. So we developed a correlation between plasmid and RNA separation based on the wash conductivity. So this really allows us to use a standard wash condition at a specific conductivity shown in the blue line in the figure on the left. And this allows us to ensure really a bulk removal of RNA during that first anion exchange chromatography step. So it really does 2 things. It ensures the RNA is being removed and ensures we're not taking a yield hit to the plasmid DNA. So we also notice that this is independent of plasmid size. So we've generated this data for multiple different plasmids of different sizes from 2.5 kb up to 8.5 kb. And we've observed a very similar trend. So on the top, the red diamonds are where you could see the plasmid DNA is being eluted at that higher conductivity. And the orange boxes on the lower part of the graph is showing where the RNA is being washed off of the column during that wash phase. So generating this data, looking at a gradient, and really establishing what that key conductivity is across a range of plasmid types gives us very good confidence that we can get high yield and remove most of the RNA via that first anion-exchange chromatography step. So regarding the hydrophobic interaction chromatography step, this serves as the final purification strategy to remove any impurities and enrich the supercoiled topology prior to the final fill and final sterile filtration. So we really challenged our hydrophobic interaction step to assess what level of supercoiled topology enrichment we could achieve for a couple different plasmids. So if you look at the table on the right, we have data showing from 3 different plasmids, a different feed material from plasmid 1, plasmid 2, and plasmid 3, ranging anywhere from 66% supercoiled up to 94% supercoiled as the feed stream entering that final hex step. And really what we're able to observe is that the final topology, will always meet the minimum spec of 85% as set out in the industry. And really, we can even do much better than that. So regardless of the incoming feed material, we're able to enrich it to about 88 to 89% supercoiled topology, really allowing a very high quality profile product coming out of that second chromatography step. Our optimized platform has led to consistent yield and quality across a wide variety of plasmid types and sizes. So this is shown in our critical quality attribute summary table on this slide here. So what we've pulled together is we pulled together a table and a subset of data for at least 10 different batches for each of these different categories. So we broke the categories into 2 different plasmid sizes, from anywhere less than 8 kb, and then a separate category greater than 8 kb. So really there are a few takeaways from this data. So one, we do see some difference in the downstream yield as a function of plasmid size. So typically the plasmids that are less than 8 kb, they perform similarly, but they're slightly higher yielding, around 75% in that downstream step, as opposed to plasmids that are greater than 8 kb, which are yielding a little lower in that downstream step, usually around 60 or greater than 60%. However, you can really see that the percent supercoiled topology is very similar between the 2 different categories. So always greater than that traditional 85% supercoiled. Here always showing a minimum of 96% or even 97% for these plasmids. So regardless of the size, regardless of the yield, we're still seeing very high quality coming out of that downstream step. And then finally, with the other residuals such as bioburden, endotoxin, RNA, host cell DNA, we're seeing values that are much lower than what the typical accepted ranges are in the US cancer vaccine guidance, Bioforum guidance, US Pharmacopoeia, and some other trade groups as well. So really indicating that we're well below what some of the common acceptance criteria is for plasmid as a starting material in the CGT space. So in general, this ensures that we can generate a high quality material due to consistent performance. It also provides a good sense to us of what our typical upstream and downstream yields are, so we can work with our clients to select the appropriate scale prior to beginning a GMP batch. So we'll have our initial R&D data. We have a good understanding of how this material will perform in our process, so we can make any changes to the scale and size up front before beginning a large-scale manufacturing run. To supplement our plasmid science and manufacturing, we also offer a toolkit of plasmid capabilities to support plasmid through the entire life cycle. It's broken into our off-the-shelf products, where we have a variety of common AAV and LV plasmid backbones. We use our plasmid science to design, construct, and test these plasmids for both high quality and high quantity vector productivity. So we offer a variety of common AAV serotypes, both single-stranded and self-complementary GLY backbones, to support customers who don't already have a selected plasmid system prior to beginning to work with us. We've also had customers that have tried out ours and compare them to their plasmids that they've used historically, other helpers or serotype-specific rep caps, and we have some good head-to-head data, as Brian showed earlier, showing how our plasmids are superior to some of the off-the-shelf solutions currently in the market. So we have greater than 20 AAV off-the-shelf plasmids, 4 lentivirus off-the-shelf plasmids, and we continue to develop and screen future generations of plasmid to produce any producer cell lines, packaging cell lines, and to really supplement any double or triple or even single transfections as future AAV space continues. In addition, we have customer services from really construct design, E. coli strain selection, process development, scale-up tech transfer for any non-platform-based projects, and any cell banking, plasma manufacturing, and the subsequent analytic and long-term stability studies. So therefore, we really offer an end-to-end complement of products in an a la carte method or a full service offering to meet our customer needs. So regardless of where a customer is in their plasmid journey, we could meet them as a one-off in any one of these different capability buckets, or we could supplement in an end-to-end design our customers' needs all the way through their plasmid service. So we manufacture plasmids with precision. This is accomplished via our 4 suites across a variety of grades, from our preclinical R&D, our Precision Pro, which is our early phase clinical, and our Precision GMP, which is our late phase clinical and commercial manufacturing capability. We offer applications from 2 liter up to 50 liter scale. We offer very consistent yields and very consistent quality, typically greater than 90, even 95% supercoiled topology, which is the main desired isoform for a plasmid used in a triple transfection process. And this is assured via next generation sequencing and any restriction digest mapping to ensure proper sequence integrity and sequencing from our material. Our off-the-shelf plasmids, our platform process, and our platform analytics ensure that SK Pharm Teco can support any customers really at any stage of their life cycle. And this is accomplished via our in-house plasmid design and screening, our in-house backbones, and then really supplementing and transforming that into our platform for our in-house method, and then offering a robust tech transfer package as well if a customer comes to us with a previously manufactured solution, or if they have a plasmid not as a starting material, but as the final drug substance or drug product. So finally, plasmids are only one piece of what SK Pharm Teco is doing to optimize the cost of goods for our customers. So we're really revolutionizing manufacturing throughout our platform approaches as an integrated service of plasmid manufacturing, viral vector manufacturing, cell therapy manufacturing, with the accompanying analytical services for each modality. We also offer both R&D, process development, and GMP manufacturing services. So again, regardless of where a customer is in the journey of their modality, we have the capabilities and can partner with you to extend your product and move it forward into its life cycle. So our toolbox of solutions offer the best quality to our customers. We do this through a variety of efforts, such as our proprietary and low-cost cell lines, our enhanced vector productivity via some of our small molecule boosters that we're testing in our process, guaranteeing performance for AAV and LV using our in-house platforms. And this is really leading to high quality, reduced costs, and we continue to make these efforts as we bring on new capabilities, continue our technological development to generate packaging, producer cell lines, and really become more efficient in our CGT manufacturing platforms. So I'd like to thank you for attending our webinar, and we'd like to encourage anyone to please contact us at the email listed below or the QR code on the upper right-hand slide, and please follow us on LinkedIn as well. So with that, I'd like to thank everyone for attending and turn it back over to the moderator for any questions.
Thank you, Brian and John, for a great presentation today. Unfortunately, that's all we have time for, but please do continue to send in your questions and we will reply via email. The webinar will be available on demand tomorrow, so look out for an email from us with the link. We'd also like to invite you to take a short survey to provide us with feedback on today's webinar, the link to which can be found in the resources on the right-hand side of the screen or in the on-demand email. Additionally, an attendance certificate is available on the right-hand side too. All that's left is to thank Brian and John once more for a great presentation, and thank you to the audience for listening. We hope you'll join us again soon.